generate mutations, fans of the X-Men. But it's probably a mistake. Anyway, but the idea was you can recruit, as Luke described, using these MS2 fused variants of AID to the genome and create a diverse population of point mutants. And so Galen spent some time engineering this enzyme to get a hyperactive variant of it. So he chopped off the normal export signal. So in normal B cells, you actually want to keep this out of the nucleus most of the time because it's actually quite damaging to have this thing bopping around removing cytosines. So it's normally exported. He chopped off the export signal, used a variant that had been identified in bacteria that had higher activity, and then fused it to Cas9. And in the end, what you could do was to actually use a guide RNA to target a population of point mutants really exactly where you put the point mutant. And what was satisfying to see is, David Liu, actually a few months before we were able to publish work, showed that you could use a different deaminase. So Luke pointed this out. So they used an APOBEC. We had been working on this for about a year and a half. It was actually kind of sad when they first published it. But I think what we actually ended up doing was quite different, and it's very interesting. So what they showed was you can take this APOBEC and get really remarkably precise conversion of a C to a T effectively with their new and improved version within like a 5 base pair window. So their proposed application was if you have a disease where a C is supposed to be a T, you can put in that deaminase and actually do therapeutic editing. In our case, what we're doing is creating a window of actually reasonably diverse point mutants with the improved version, about 100 base pair window of the guide RNA. And in fact, we get C to basically everything, G to basically everything, and even some measurable levels of A's and T's being converted. So you can generate a quite diverse library of point mutants. Exactly where you put the guide RNA. So this was kind of nifty because now you have this population of mutants and you can actually start to do the type of classical genetic screens in a way that I described on the first slide, where you create a population of mutants and now you can ask, what is the thing that gives me a new function? So one of the initial kind of applications that we thought about was to do evolution of GFP, because we know exactly what turns wild-type GFP into eGFP. It's brighter, and the mutations that do this are known. So we put guide RNAs around this locus, and GFP would cause mutants to be formed, and then we sorted for GFP variants that had brighter fluorescence by flow cytometry. And after one round of sorting, we could already recover this known S65T mutation. It's a G to C, which is what's responsible for converting wild-type GFP to eGFP. So it's actually, of course we knew what should happen here, but it was satisfying to see we could mutagenize this population and recover a variant that gave us a new function. The more interesting was to do a tiling mutagenesis of PSMB5. So this is a proteasome subunit and is the target of this chemotherapeutic drug bortezomib. And so what we did was we took a library of guide RNAs targeting the entire protein coding region. We introduced this mutagenesis machinery, and then we pulsed with this drug bortezomib and asked for cells which now become resistant to this chemotherapeutic drug. And what you can see is we could recover in a PSMB5 targeting library, but not in a library targeting these safe regions in the genome, mutations that confer resistance, which was actually quite nice to see. In fact, what was interesting is this exon 3 is actually not expressed in K562 cells, and we saw no mutants coming up there. So when we looked at where these mutations map onto PSMB5, so fortunately the structure of PSMB5 is known, its structure in complex with bortezomib is known, and previously people had identified in resistant cancer cells mutations in the binding pocket of PSMB5 for bortezomib. And so we were happy to see that we could actually recover those same mutants that sit right in the binding pocket. But we could also identify a number of other mutations that occur even on the complete opposite side of the protein. So would not really have been predicted from the structure, but in fact clearly affect the binding somehow of this drug for its target. And so we're really kind of excited about using this to map drug-protein interactions more generally. And it's, you can imagine using this type of mutagenesis for a lot of different applications. So the Liu Lab is doing these sort of therapeutic editing applications. We're envisioning more evolution of protein function, potentially mapping of drug-target interactions. Another interesting thing we observed with this technology is you can actually cause mutations not only in the coding region, but actually upstream of the transcription factor binding site, of the transcription start site. And that's potentially interesting because previously AID have been thought to require transcription to induce mutations. But what we could show is that as far as 3 kb upstream of the transcription start site, we could put a guide RNA in this mutation machinery and still observe mutations. These are here in yellow. So we think that this might also open up the space of mutagenizing regulatory elements that are not actually transcribed to try to find, for example, promoter variants, transcription factor binding site variants that would affect binding activity. So we're kind of excited about the different applications for this, generation of different antibodies, evolution of enzymes and aptamers. And the nice thing about this system is you really only need a guide RNA, but you can generate a diverse library of mutants by effectively doing a screen by tiling the protein coding region. And I didn't mention this, but of course you can multiplex this by just targeting multiple sites at the same time. So you can express 2 guide RNAs in a cell. You can co-evolve 2 sites that would potentially have some new function if mutagenized together. Okay. Does anyone have any questions so far? Otherwise, I have a couple of quick points on kind of retesting hits I can go through. Yeah. So not too much with the AID system. Yeah, so you do see some basal level that's slightly elevated, but really, you know, if you look like, really not worth going all the way back to make this point, but anyway, you can see in this case, for example, by and large, you really only get mutations where you put the guide RNA. So yes, there's some like infinitesimally elevated level if you just do, you know, whole genome sequencing, for example. But you know, at the level of, you know, substantially enriched mutations, it's really not detectable outside of where you put the mutations. Yeah. Okay. So the last 2 minutes, I wanted to just describe a couple of other strategies for kind of following up on hits from screens. So one of the things that we found to be useful, you do a whole genome screen, you may not want to clone 100 different guide RNAs against your 100 top hits to actually see which of these are actually real. And so one of the things we found to be useful is to generate kind of a batch library. Here's for shRNA, but the same thing has been useful for guide RNA libraries. If you do a whole genome screen, you can now generate a mini library, for example, of your favorite, you know, 100 genes, and retest them at much higher coverage and get much higher quality data. And that allows you to really more efficiently process through the hits to get the effective ones to follow up on. And of course, you can do this genetic interaction map strategy. We've also found these competitive growth assays to be quite useful. So you can infect a cell with a guide RNA targeting your favorite gene and a fluorescent marker, mix them with the wild type population, and then study the behavior of those populations in a mixture. And you can, you know, for example, if this gene causes drug resistance, you expect it to take over and become, the population should be 100% green if you start out from a 50-50 ratio. So this is another strategy we've used to kind of follow up on hits from these screens. Of course, you can tag genes. I think you guys are probably going to be talking about gene tagging and gene knockouts in some of these workshops, but this is a gene, we had no idea what it did from a screen. You can use CRISPR to knock in GFP and actually localize this thing in the genome and see where it goes. And of course, this is greatly facilitated by these RNP studies, which I think you guys are going to learn about in the afternoon. So some of these new tools really make tagging a gene with GFP much easier. So that's pretty much it. Some references, a lot of people to thank for the work here. A lot of the early work was done together with people in the Weissman lab, including Luke, Max, and Martin, and people in my lab who did the work that I showed you, Richard, David, Michael, Edwin, and Galen, and Q. So I'll take any other questions you guys have. So we think we can see mutations even upstream of a transcription start site. So we haven't tried long non-coding RNAs yet with this thing. When we've tried to put it in a completely dead region in the genome, you know, transcriptionally off compact chromatin, we really don't don't see any activity, so there probably is some requirement for at least accessibility. We don't really know yet how much transcription is required. Even at the promoter that I showed, where we can put a guide 3 kb upstream of a transcription start, still see mutations. I can't guarantee you that there's zero transcriptional activity at that spot. There may be some transcription going in the opposite orientation. I would expect if the non-coding RNA is transcribed, you'd have a decent chance of mutagenizing it, but we haven't tried it.
That I'm going to tell you something that's interesting, and of course I have this very biased view, which is I look at things from my computational lens. And are there any computer scientists in the room? I was anticipating not, but okay, there are. So there's one. Maybe every now and then I'll ask you a question. No, I'm just kidding. But so my goal here is going to be to basically actually just give you a flavor of what is machine learning. This is my expertise, and so just actually again to get a sense of who's in the room. Like if I picked on someone here, like raise your hand if you would be able to answer that question. Like, what is machine learning? Okay, a handful. No, actually one or two. Great. Okay, so I just want to give you a sense of that, and I'm gonna, most of this is going to be pretty intuitive. I'll try to make little bits of it concrete that I think will be helpful, and then I'll tell you how we used machine learning to improve guide design, specifically for knockdown experiments. But I think a lot of it is probably useful for more than that, but we haven't sort of gone down that route, and so I can't say very much about that. And please interrupt me if something doesn't make sense or you have a question. I'd rather do that so everybody can kind of stay on board rather than it makes less and less sense the longer I go. So machine learning actually during my PhD, one of the big flagship conferences was peaking at around 700 attendees. And when I go now, it actually is capped, like it's sold out at 8,000, like months in advance. So because this field is just like taken off, basically it's now lucrative for companies, and it's become a really central part of Google, Microsoft, Facebook, and all the big tech companies. So this field has changed a lot, and kind of similar to CRISPR, there's an incredible amount of hype and buzz and ridiculous media coverage, and so it's a little bit funny, in fact, that I'm now working at these two kind of very hyped up areas. But anyway, so people in just the mainstream press now, you're always hearing about artificial intelligence and deep neural networks, and so these are I would say machine learning is a sub-branch of artificial intelligence, and a deep neural network is sort of an instance of machine learning. And so what really is this thing? So it kind of overlaps sometimes with traditional statistics, but in terms of the machinery, but the goals are very different. But really the core fundamental concept here is that we're going to sort of posit some model. So maybe think linear regression is a super simple model, and you can expose it to data. It has some parameters, right, the weights, and then we essentially want to fit those weights. And that's the training. That's literally the machine learning. So I'm sorry if that sounds super simple and not godlike, machine learning and everything working magically, but that's really what it is. And right, and so let me just also give you sort of drive home that point. So we're going to posit some sort of model, and so here I'm giving you the simplest example because I think most people here work with linear regression at some point in their life. And so you can think of this as a predictive model in the sense that if I give it a bunch of examples of y and x, and I learn the parameter beta, then for future examples where I don't have y but I only have x, I can just compute x times beta and I get a prediction of y. So that's the sense in which I call this a predictive model. And that's very much how machine learning people tend to think of it, where statisticians are often very focused on what is beta, what are the confidence intervals around beta, and things like this. So that's the sense in which there's a lot of overlap, but the goals are kind of quite different. We want to use real data and make predictions, so here it's going to be predictions about guides, and which guides are effective at cutting and at knockout. Right, and so it has these free parameters, and we call these things that we put in here features. And so in the case of guide design, the question is going to be what features are we going to put in there that allow us to make these kinds of predictions? And so I'm going to get into that in a little bit, but just as an example to make this concrete, it might be how many GCs are in this 30-mer guide, or guide plus context. Right, and like I said, we're going to give it some data, and so in this case, the data for guide design is going to be data from John Dent or others in the community that's now publicly available, where there are examples of, for example, what the guide was and how effective the knockout was, or what the cutting frequency was, for example. And I'm going to get a bunch of these examples, and then that's going to enable me to somehow find a good beta. And of course, we're not, actually, we do sometimes use linear regression, but I'll tell you a little bit more about more sort of complex and richer models that let us do a lot more. And then the goal is going to be to fit this beta in a good way. And like, I'm not going to do some deep dive on that here, but the one way that you are probably familiar with is just mean squared error. And when you find the beta that minimizes this for your example training data, then you get some estimate of beta, and you hope that on unseen examples, when you do x times beta, it gives you a good prediction. So does that sort of make it somewhat concrete what I mean by a predictive model, and how you could view linear regression as a predictive model, and how you might use this for guide design? Okay, so obviously I'll tell you a lot more. So right, but linear regression is just sort of the simplest possible example. And so in our work, we actually use some of the time what are called classification or regression trees. And so in contrast to here, where you might have, say, you might have a bunch of these features, right? Like how many GCs were in my guide, and then another feature might be, was there an A in position 3? And you can put in as many as you want, and then you get all of these betas estimated. So it's very simple, because in that case, none of these features can interact with each other, right? You just add x times beta 1 plus x times beta 2. So we call this like a linear additive model. In contrast, these trees allow very sort of deep interactions among the features. So this might be how many GCs. Of course, this is just, this is not suited to the features I just described, but this might be some feature like, I don't know, proportion of GCs, because now it's fractional. And then this algorithm, which is going to train the betas, so find a good value beta, will sort of, through a procedure that I'm not going to go into detail for all these models how it works, but it's going to somehow look at the data and determine that it should first split on the second feature at this value, and then it will sort of keep going down that. It says now partition the examples in my training data like this, and then on the second feature in this way, until you end up at the sort of leaves of this tree, and these leaves are the predictions. And so when you do it for the training data, whichever training examples here end up at this leaf, you basically take their mean, and that's now the prediction for that leaf. And if you take a new example, you basically just pipe it through these sort of rules, and you end up with that kind of a prediction. It's simplified, but I think it's a good conceptual view. And this is just another way of thinking about it, is if you only had two features and you drew them like one against the other, then effectively every time you make a branch here, you're kind of cutting up the space. So that's also just another way to think about it. And so also, so this is, all over the press nowadays, and whenever I give these talks, there's a bunch of young, hungry grad students who say, did you do deep neural networks? That's what everybody wants to do now. And so deep neural networks are kind of like a really fancy linear regression. So you can think of these as the x's in linear regression. You can think of this as imagine there's only one thing out here. I should have done a different picture. That's just y. And so this, again, is a mapping where you give it the x's, you do a bunch of stuff, and out you get a y here. Except linear regression, is this very simple thing. And now you can see there's all these other kinds of, we call these like hidden nodes, and so there's this complicated mess now of parameters beta. And again, I'm not going to go into it. I just want to give you a sense that like linear regression is this very simple thing, and there's a lot of other models that let you do much, much better prediction. And these are typically the kind of models that we use because they're more powerful if we care about prediction. But the flip side is they're actually very hard to interpret. And so if you want to ask a question like, was it the GC feature, that is most important in your guide design, which is what I always, get a question like this. And we can do our best to kind of poke and prod at this machine, but it's always a little bit ad hoc. It's hard. The more complicated the model, then the better we might predict, and the less interpretable it is. And so there's always this kind of tension. So right, and so what are some of the challenges? So I've sort of shown you sort of some like increasing amount of complexity in some models. And so one of the big difficulties is if I posit a very complex model with a lot of parameters, then I need a lot of data in order to actually fit those parameters. And if I don't have enough enough data, what's going to happen is the parameters, you're going to find this very specific setting of the parameters that effectively memorize the training data. And the problem then is you give it a new example that you really care about. You say, I want to knock out that gene, and it's never seen that gene. It's because it's sort of memorized, what we say it's like overfit to the data. It doesn't actually generalize well to these unseen examples. So there's another tension here, which is you get all kind of complex, rich models that can capture very complicated prediction spaces, but if you don't have enough data to warrant them, then you're going to shoot yourself in the foot. And so, when you learn in this area as like an undergrad or grad student how to do rigorous machine learning, this is one of the things you need to understand, how to control that knob, how to know if you have enough data for the model you're looking at, and things like this. And so in this case, this is kind of fun for us to do the guide design, because a lot of people are using this now, and we know we didn't super overfit it because people are telling us that it's useful. But we did a lot of due diligence on our end to convince ourselves that that was correct as well. Right, and even just asking, how can I evaluate a data set? There's a lot of papers out there where people sort of dabble machine learning, they go use something, and they prove that they have this amazing predictive capacity. And in many cases, because it's such a popular area now, people just kind of jump in and they do something, and these evaluations are actually not rigorous. And so you need to read things very carefully. You can't just look at a result the way you can look at a gel. Maybe I'm wrong here, correct me if I'm wrong. If it's not fabricated, then it's not just what you see is what you get. You need to think very deeply about what they've done and if that makes sense. Right, and then of course, like in linear regression, for example, we assume that the error term is, for example, Gaussian distributed. So this is like a very sort of technical, precise thing. Of course, almost in most cases, and every model has some sort of assumption like this. And basically, when you're working with real data, every model is wrong. Like there's no way. I mean, the real model is physics, right? And we're not using physics-based models. They're hard, they're not scalable, and basically it's almost unheard of, except in a few select fields of molecular dynamics. And so again, this is one of sort of the tricks of the trade, is how do you, and to do it rigorously, is how do you know that this assumption, even though it may be violated, that your model is still okay. So that's another consideration. Sometimes we think it's okay, but maybe if we can violate it less by doing something different with the data before we give it to the model, maybe we can do better. And then in a lot of the work I do, sometimes exactly a very good model, but it's just really, really slow. And so you need to be clever about actually how to speed things up. And it might be slow when you're trying to infer those parameters during the training phase. So I often refer to like a training phase where you have this data, and then there's sort of a deployment or test phase where you're just using the model and making predictions, but you don't know the right answer unless you're evaluating. So that's sort of, I guess, just a sort of overall little bit of a view of a slice of machine learning. Are there any questions so far? Okay.
One of the challenges, of course, is you have to have a handle on all the features that are important.
Yeah, absolutely. And so in a way, one of the sort of miracle breakthroughs of deep neural networks, and that that's touted, is that you don't need to know them, because it sort of is such a rich machinery, it can create features on the spot. But the problem with them is it needs a vast amount of data to do that. And the other problem is they've really only been shown to be state-of-the-art. They've broken 30-year benchmarks in on things like speech processing and vision tasks, which are images and speech, which have particular properties like continuity in time for speech, continuity in space for vision. And people are applying them everywhere now. But in many cases, people say, I used a deep neural network, but you know what? If they just used some simpler model, they would have done just as well. But that is sort of, so what you said is exactly right. And when you don't use deep neural networks, and even then sometimes if you don't have a lot of data, then yeah, this sort of feature engineering is what they call it, is what do we put in? Do we give it just the GC content? Do we give it this? Well, kind of one of the beauties of machine learning is that you can posit a whole bunch of things, throw them all in, and if you have enough data, then it can tease apart what is relevant and what's not. But again, there's always this tension of if you give it too many things and you don't have enough data to warrant it, then you're going to get a bit of nonsense out.
true that you still can't look under the hood and see which
Yeah. So this is a very active area of research, although I would say it was a bit obscure up until very recently, and the reason it's become much more important is because deep neural networks are so important, and people are using them for problems that, like, you know, physicians are, like, looking at a CT scan and stuff, and medical areas especially, people want to know, well, why did it make that decision? Like, I'm not going to give my patient this or that or not do that because your weird, crazy deep neural network just popped out 0.8, you know? And so this is a very active area of research, and there are things you can do, but it's I would say it's always, in some sense, an approximation over what, because at the end of the day, it's something it's like, you know, a human works as a whole, right? And then you ask some very specific question about some individual thing. Well, you know, like in biology, we ignore like the whole rest of the system just to try to understand because we don't have a choice, right? And it's very much like that in machine learning as well. So you do ask and you get something that's useful, but you know that by ignoring every other system and every other thing that you can only go so far. Right. And so I just want to give you also a little bit of a flavor of sort of what I would call the two main branches of machine learning. And everything I've just told you about is what we call supervised learning. And so in supervised learning, why is it supervised? It's because in the case of like guide design that I'm going to tell you about, the way we do it, the way we build this predictive model is I give it a supervisory signal in the sense that I give it a bunch of guides, and for each guide I know how effective the knockout was. And that's the supervisory signal. And so without that, I mean, in a sense, how could you make predictions, right? You can't build a predictive model if you have no examples of how well it's worked in the world. And the trick is to take some limited number of examples and do something that generalizes to a much larger set of data. And so that's and this is, you know, obviously used for any number of problems in biology, outside biology, in finance, like anywhere in chemistry, you name it, this exists. And so my own background, I should say, is a little circuitous. Like my undergrad was actually in physics, and then I actually did computer vision, and then I got into computational biology during my PhD, and I've meandered around like through sort of immunoinformatics, proteomics, and so in fact these are kind of problems that I have looked at, and now more recently I'm doing some of this CRISPR guide design. And so, right, and one distinction also is, am I predicting a yes-no, like knockdown or knockout versus not? That's classification, because you have a class of yes it was versus no it wasn't. And regression is sort of like how much was it knocked out, or like, you know, what fraction of cells, or what was the cutting frequency, something that's not discrete. And we don't need to pay a lot of attention to that, but in our papers on this topic, we have shown that people like to view this problem as 0, 1, like it, you know, cut or it didn't cut. But if the assays that you guys produce, like, give much more information than that, and it's kind of crazy to just ignore that. And therefore, we want to be in a regression setting where we're actually predicting real values. And, you know, mostly we care about the rank, because the kind of scenarios that we've been trying to tackle are, someone says, I want to knock out this gene, so we want to go through, and I should say everything we've done here is also for Cas9, just like the wild type Cas9. And so we want to, you know, we say, our goal is to essentially say, rank all the possible guides for that gene for Cas9, and then compute the on-target according to our models, compute the off-target, and depending on your use case, you balance those in some way that you think is appropriate. And so we want rankings here. Like, we don't care if we know the exact number, although I guess some of our users say, like, well, what does that number mean? And that's very hard to do because of the nature of the data that we take in and the sort of different assays that get incorporated and how we have to kind of massage the data to throw them all together, which I'm not going to talk about here, but is a very difficult problem, actually. Right, and so in this setting, it's pretty easy to evaluate the model. It's not trivial, but it's much easier than the second setting, which is unsupervised, which I'll tell you about in a second. And the reason it's easy is because I have some set of examples for which I know the answer, right? And so the kinds of things that we do is we basically fraction off the data. We keep some amount of our data away from us, and we just never look at it until the very end. And then we do a bunch of our own sort of methods on this other part, and we are allowed to look at it as many times as we want, and at the end of the day, then we go kind of once to that final validation set and say, how well did we do? And we can do that for different methods, right? So we can look at competing methods from the literature or competing methods we make up and say, like, okay, well, is this, you know, how do they fare? And so in that sense, it's like, that there can be problems with that, but it's much easier than in this case. And so, This is unsupervised learning. I'm not going to talk anything about that other than this one slide here today, but I just wanted to, if you've heard this, thought it might be useful. So probably everyone here at some point in their life was doing something with gene expression, and you would draw these little plots from, I think it was from Berkeley, right? Wasn't it Eisen, Mike Eisen? Okay, maybe this is a different, I don't know, who here has drawn these plots at some point in their life? Okay, at least more than people who could define machine learning. So, right, so one axis here, or I can't actually see, what is it? Yeah, so this is, each of these would be a microarray experiment, every column here, and then people would say cluster these for, people with colon cancer or something, and then they'd say, oh, are there subtypes that we can see by clustering them according to this tree, and you would sort of cut off the tree and give you some clusters. So in this case, the sort of analog in guide design would be, I give you just the guides, but I don't tell you anything else. Like, I don't tell you how efficient the knockout was at all, and then so you're just trying to understand some properties of this set of guides. And for guide design, this, as far as I know, this is not interesting or useful. I don't know what you would do with it, but in other cases it is. And I've spent a lot of my life actually in statistical genetics, where you can do some really cool stuff. So when you're doing GWAS, like genome-wide association study, and you're scanning through looking for each marker, is it sort of related to this phenotype or this trait? One of the things that really messes you up there is when you have heterogeneity of, say, populations from the world. And so one thing people do is they kind of do this unsupervised learning. In this case, it's called principal components analysis. You can take all of the genetics and you do this process, and then say each person in this room, like say we've measured, you have 23andMe, whatever, you have all your genetics, and then you can do this unsupervised PCA, and now each person will get 2 numbers out of this analysis, this unsupervised analysis, and you can plot them here. And when you do that, it actually kind of recapitulates the map of Europe if you happen to know where you're from. So that's not going into the algorithm. And so that's kind of cool. I mean, I haven't told you exactly why we want to do this, but it's just kind of a super neat result and totally irrelevant to this talk. Right, so that's my sort of very quick and rough intuitive intro to machine learning, and I'm going to jump into how we use machine learning for the on and off target problem in guide design for knockout. But again, are there any questions at this point? Okay. Right, so this one, the first one I'm going to talk about is on target, and that one's already published. This is with John and other folks at the Broad. And then this one is in revision now, although it's on bioRxiv, not the most recent version. And of course, our overall goal was to have some sort of end-to-end guide design service, but we got to do this in pieces. There's almost no end to this, right? Even when we have our current one, there's going to be a million things we can do. And I think so I actually often talk more to computer scientists, rarely to biologists, and I have to explain in great detail what it is we're actually doing. And I suspect everybody in this room knows, but just to be clear, again, I'm going to focus on the knockout. Although often the assays and the modeling we do are actually cutting frequency. So in this case, I'm considering knocking out this gene. I'm going to use Cas9, and maybe there's like NGGs at these 4 positions. Of course, it's more than 4. And the idea is I want the one that's going to most effectively knock it out. Or I want to rank order them in some way, right? And then let the user decide what to do. And so I want the best guide and to guide me to the good position. And then, of course, the flip side is if I've chosen that particular guide, then I want to make sure I'm not disrupting the rest of the genome. And so we model these separately. And now I'll go through one at a time. Right, so this is a little schematic of what I mean, which I've been alluding to, I think, in the introduction about how we're going to do guide design. So again, this is going to be supervised in the sense that John's going to give us a bunch of examples in his wet lab where he's measured the knockout efficiency. And what I'm going to do is I'm going to take the guide, and it's actually, we have a 30mer here, so it's 23 plus I think it's 4 extra on one side and 3 on the other. And to be honest, like, that's not something we ever played with much. Somehow I think we just started using 30. Maybe we tried just the guide and this helped. I don't actually remember right now. But it's a 30mer sequence, and then we're going to learn some function. And so again, in the simplest case, this function might be linear regression, where you're effectively just learning some betas. And then it's going to allow us to make predictions. And the game is going to be to collect as much data as we can and to fit these in a suitable way. To pick a good model and then to fit it in an appropriate manner. So that's the setup. And so these, I've just drawn here kind of 3 of the key components, the decisions you have to make when you're doing this kind of thing. So the first one, and which Dana just was mentioning is like, what features do we give it here? Like somehow I go from this, ACTG guide to some numbers, and I'll tell you a little bit about that. And then the other thing is we need some measure of the supervisory signal, is how effective it was. And so that's not, as a computer scientist, that's not in my control, but John and you guys are super clever and managed to figure out how to measure these things. And then our job is to think about, when you measured it for that gene and that gene, are those comparable? And can we throw them together or not? And things like this. Or should we, transform it in some way so that it better adheres to the model that we're trying to use, maybe Gaussian noise and things like this. And then finally, what model are we using? Or are we going to try a bunch of models, and how are we going to decide which one is best? So John, in this first paper, he had some data already there, and then he gave us some more, and overall we had, I think I have a slide on this, it was for just 15 genes. He did systematically every possible place you could deploy Cas9. Okay, great. So, he had this one he'd already published, which is they were all cell surface proteins, and so you could use flow cytometry and fluorescence to separate them and get some measure of knockout efficiency. And then the second class of genes was using a drug resistance assay, where I guess it's known, and I guess they showed in the paper as well, they reestablished that when you apply this drug, and you successfully knock out the gene, then the cell survives, and otherwise not. And so using these kinds of